So thanks again, everyone, for being here. I've been asked or given this opportunity to give sort of an overview. So actually the idea is to give a talk that introduces the field to people who are not in the field, especially, say, grad students who are in physics and math, who want to see kind of what is the workshop about. And I have to admit, it's quite a broad field because there's so many different areas of biology where we find these extremal statistics matter. And then even in math, it's a very broad topic that connects to different things in statistical physics. And so I haven't covered it all, but what I tried to do is just pick examples that are representative of how we encounter extreme statistics in biology and sort of point out some of the common threads. Okay. And the talk is supposed to be recent developments, but we're going to start in the 1940s. So not so recent, but I think this is, if you want to talk about extreme statistics in biology, this is a pretty good place to start. So it's the 1940s, and people, they understand natural selection, they understand their genes, but really have a pretty rudimentary understanding or not much of an understanding about sort of the molecular basis of mutations. And in particular, there's this question about are mutations, when a population is exposed to selection pressure, are the mutations induced by the selection pressure? Is the selection pressure causing the mutations, or are they pre-existing in the population? It's a distinction that was made between sort of this view of Lamarck and Darwin. And so basically what people are debating is basically the picture on the left. So this is a population growing. Each branch is a cell division. And it's a very simple, nice population where the divisions are perfectly synchronous and everyone's growing at the same rate. And on the left, what happens is at the end of the experiment, they're exposed to a selection pressure. And all of the cells in red are the ones that get a mutation in response to that selection pressure. And so those are the ones that survive. Whereas on the right, what's happening is the mutations are, as we know is true now, they're pre-existing in the population. And what these two guys, Luria and Delbruck, are trying to do is they're trying to tell this apart. And it's maybe not so obvious just from looking at this picture that if you didn't have access to this tree, if you were just doing these many replications of this experiment of growing cells and counting the ones that survived at the end, it's maybe not so clear that you could tell these two apart, but it turns out that you can, and they did. And this led to a whole kind of lineage, no pun intended, of mathematical research. And in fact, even predating Luria Delbruck, I learned while making this talk, there was this guy Ewell who had actually been thinking about this very problem. It kind of had a different motivation for it, but had anticipated this very situation of trying to understand the distribution of mutations at the end, the number of mutations at the end of a population that was being grown, where those mutations happened randomly throughout the tree. And there was, I'm just mentioning some of the work that followed up on this. Lee and Coulson were famous for figuring out some of the formulas that are now used even still to determine mutation rates. And then there's a lot of modern work on this problem. So it is so interesting just trying to figure out that distribution that people are still thinking about it, still working on it. And what I want to point out in particular and talk a little bit about in the next couple slides is that that Mandelbrot was really the one who took this problem and connected it to a separate body of work, separate at the time, in extreme value statistics and probability theory, where people were thinking about things like heavy-tailed distributions in extremal statistics. So what does the math look like that kind of goes into this? And I'll only go through it pretty briefly, but basically the idea is that if I'm thinking about this problem in the biologically relevant regime that Luria and Delbruck were interested in, the mutation rate's small, and so you can basically treat all the progenies of each of the mutations as independent of each other, and you really just need to add up the total number of sizes of all the progenies. So all those kind of red subtrees that protrude from the mutations, right? The red ones are the ones that get the mutations. And so how do we figure out the distribution of the sizes of those subtrees? Well, if you think about a mutation that occurred at time tau back in time from where we currently are from the end of the experiment, right, it's going to give rise to 2 to the tau cells at the end of the experiment. That's going to be the progeny size. And so to figure out the distribution of that tau, you basically just need to look at, okay, what's the the branches before n minus t, if you're looking at probability tau is greater than t, the fraction of branches before n minus t, n's the size of the tree over the total branches, and you can work that out very easily, and you get this exponential decay in tau. So you're more likely to get the mutations at the end of the experiment because you have many more branches there. And now, if you go to look at y, the distribution of y, and you plug in that distribution of tau, the exponential decay of the time to get the mutation kind of cancels with the exponential effect of having a mutation early in the experiment. So you're exponentially less likely to get a mutation early, but it has an exponentially large effect, and boom, this kind of interplay of exponentials gives you a power law. And if you had done this with a more complicated tree where things were kind of, it wasn't synchronous and the fitnesses were different, you could get a different type of power law for that distribution. And this power law was probably, this was why Mandelbrot was interested in this. If you've heard of Mandelbrot, if you know Mandelbrot's name, you probably, and you don't know about Lauria Delbruck, or you're not into financial mathematics, you may think of one of these pictures. It comes from complex analysis, this Mandelbrot set, and it has these kind of, it's much more complicated, but it has these scale and variant properties where we sort of zoom in and we see similar structures on different scales, and that's the same property that we get with power laws, where we kind of rescale the y by some power of n. We can retrieve that by dividing the original distribution by n. So that's kind of the like a basic picture of the mathematics that went into this, and it leads us to think basically about sums of random variables that have power law distributions. And this goes back to long before Mandelbrot. When Mandelbrot was doing his work, Paul Levy had kind of laid the foundation for thinking about these kind of sums of power law distributions. If you've heard of Levy processes, that's related to this. And by the way, I was not able to find any evidence that Lauria Delbruck knew about Levy's work. They had actually figured a lot of this out with kind of more elementary techniques. They were able to figure things out, like the variance of the distribution and formula for the mutation rates. And later, so if you get into this literature, some names you might hear are Frichet, Zhengenko, Kolmogorov. They were thinking about these sums, and they kind of realized that it's really the order of the maximum element that's determining this distribution. And then another, I'm just kind of throwing names, give you a little bit of a picture of the history of this field. Fisher and Tippett were sort of, did the early work studying probabilities of extreme statistics. So they were thinking about the distribution of the maximum of a bunch of independent random variables. And if you do this with power laws, what you find is that the max, well, it also has asymptotically its power law form. And if I look at the max of n independent power law distributed random variables, approximately I'm going to get n times y to the negative alpha if it's decaying as a power law with exponent alpha. And that means that if I want this thing, if I want to see the probability to be order 1, I should plug in for y something that's on the order of n to the 1 over alpha. So that's basically saying that I'm likely to see in a sample of n power law distributed random variables something on the order of n to the 1 over alpha. And Fisher or Zhengenko and Kolmogorov took these early results, and they proved a theorem which is a generalization of the central limit theorem, which you all know. So they're thinking about this problem of if I have a sum of independent, identically distributed random variables, I want to find some rescaling of it, some shift and some multiplication of it, so that in the large n limit, it converges to some distribution. And we know that, okay, that's the central limit theorem if the variances are finite. If the variances are not finite, what's going to happen? Well, it turns out you can still do that. You get something different. It's called an alpha stable distribution. And actually, the normals example is also a stable distribution. And you can go through the case where if alpha is between 1 and 2, that's going to be the expectation is finite, but the variance is infinite. And you can do the boundary case too, but I'm not just, there's the like logs there. But you can work that out too. And so in this regime, the thing is that the max, right, in the central limit theorem regime, the max is growing slower than those square root of n fluctuations. So that's kind of saying, okay, not really that important. But when you're in this regime, the max is growing faster. And so you don't have a defined variance, and it's that n to 1 over alpha that's determining the scale of the fluctuations. And you can keep going. You can do the same thing when alpha is between 0 and 1. And basically, there's these stable distributions have kind of a nice form for the characteristic function, which is the expectation of e to the I of that variable. Now, I'm just showing a picture here of what to give you kind of a visual idea of what it looks like. So these are the running sums in the different cases. And again, when alpha is greater than 2, that's kind of what we're used to, the central limit theorem regime. It's just growing linearly. When alpha is between 1 and 2, it's still pretty much going to grow linearly, but with more fluctuations. And you're always, with the meaning of this infinite variance, you're always likely to see a y that's kind of bigger than all the other y's you've seen with like a order 1 probability, where when alpha is less than 1, you're likely to see a y that's bigger than the whole sum itself, and it sort of kills that smooth growth. And so they worked out all these different regimes, and it turns out you can do something similar for the distribution of the max. So the max is important for this, but you can also look at the distribution of the max, and that leads to these Gumbel, Fauché, and Weibull distributions. So we've already met some of these this morning. And a lot of interesting connections. I'll talk more about this at the end. There's connections to physics and renormalization group approach to these problems. Ariel's done some work on. And there's also this idea, I won't really get into it, but this is a part of a more general theory of infinitely divisible distributions, which is essentially you can look at the limit behavior of a sum where they're not power laws, but they still have undefined variance or undefined mean. It tells you what you can get. And of course, I just mentioned it circling back to Lauria-Delbruck, indeed, you can show that in certain limits, this Lauria-Delbruck distribution is well approximated by one of these stable distributions. Of course, there's much more to the Lauria-Delbruck distribution, though, and it's kind of subtle how you do the limits, how to actually get that. Because remember, I'm taking like a large population, but I want to take the mutation rate small. So there's a lot more to Lauria-Delbruck than just stable distributions, but it's kind of hidden underneath there in these stable distributions. So the key thing I want you to take away from this part of the talk, because I'm trying to emphasize when do we care about extremes in biology? Why are they pervasive? And in this case, in the Lauria-Delbruck experiment, it has to, again, to do with the fact that you have something exponentially rare that has an exponentially large impact, okay? And that's kind of a generic thing, especially if you're dealing with growing populations. That's kind of the defining feature of biology in some sense. People argue about this, but, self-replication. So you have self-replication, you have exponential growth, and things get amplified. And so a natural question you can ask is maybe what are the implications of these kind of, if we expect generically to get heavy-tailed distributions, what are the implications of that more broadly for evolution? If you're producing heavy-tailed or power law-tailed distribution offspring numbers, what are the implications of that? And that's something that has been explored in the context of models of longer-term evolution. These models are going back to Wright and were later sort of generalized and brought more into the mathematical probability theory literature by Cannings. And in these models, essentially, unlike this branching exponential growth model I showed before, I have each generation, I have each cell, each of these is a cell, so this is like a simple model of evolution with no sex or anything. They produce a bunch of offspring, so those lines are the offspring, and then I'm resampling them for the next generation. I've just colored them so you can see how the frequencies change. And so in these models, you can ask questions about, okay, the fluctuations in those frequencies, or you could sample individuals at the end and ask questions about the structure of those genealogies. How fast do they coalesce? And this has also led to a lot of extreme value theory. I'm showing two pictures, one with the Poisson, one with the power law tail distribution for the number of offspring, and you can see the difference both in the coalescence and the fluctuations being much larger in the power law tail one. So, okay, do we see these? Is there evidence for there's actually like these power law tails in real biology? So with that, if those offspring distribution, like the case on the left that I showed, if those have a finite variance, we expect sort of square root fluctuations, square root of n fluctuations, n being the number of cells in the frequencies as this population evolves. But there's a lot of examples where you don't get that, and this is one I like. So there was this guy, Hedgecock, he was looking at lobsters, and he found that, he did some analysis of data, of genomic, of population genetic data, and he found that, okay, this like, basically these fluctuations, what they call the effective population size, is on the order of like 10 to the 4th. But fishermen, if you look at how many lobsters they're bringing in, it's like 10 to the 7th. So clearly the fluctuations are much larger than you would expect from the population size. And this led to a lot of, inspired some of this, and that like incorporating these heavy tails into these population genetic models. And then this is another example, which I think we might hear about it more later in the week, so I won't say too much about it, but if you look at COVID cases, these COVID cases from the UK, and if you look at the inferred, again, you look at the fluctuations in the frequencies of the different strains of COVID, that's suggests a much smaller population than the actual case counts, what you would infer in the prevalence of those strains from the case counts. And so, again, I think we'll hear more about that later, but this is another example where you get these sort of heavy tails. In this case, it's more like these super spreader events. In the lobster case, it's because they just produce large variation in the size of their offspring pool. Now, another getting into like what's kind of the mathematical literature that has been inspired by this, by these sorts of biological context, and how's it related to Lauria-Delbruck? Well, Lauria-Delbruck is looking at the summing up a bunch of power law distributed random variables. These problems I'm mentioning are actually about the ratios of the power law distributed variables to the sum, because at each generation, right, you're producing power law distributed offspring sizes, and then you're sampling them. So what you really care about is the fraction, right, of the pool that each individual contributes to. And that's a kind of a different mathematics. There's like different math there, but it's also very closely related. So Kingman, just to go a little bit through very briefly through the history of this, so Kingman was sort of responsible for basically proving what I like to call, although some people don't like this way of saying it, but I like to think of it as almost like the central limit theorem for these coalescence, for these genealogies. He's asking, what's the limiting genealogy that you get when the offspring variance is finite? And that's this, it's called the Kingman coalescent. And in that case, if you look at the genealogy in a very large population over a very long time, you're going to find that the branches always only merge two at a two. So you only get binary mergings of branches. And other people, notably Jason Schweinsberg, have studied this sort of, I guess what you could call like the generalized central limit theorem for these coalescents, and shown that you get these things called beta coalescents, kind of playing the role of stable distributions in the theory of coalescence. And then there's sort of work on forward pictures. So Oscar Halachek has done quite a bit of work on this. And one of the things he's shown that's really quite interesting is that when you have these heavy tail offspring, the dynamics of the allele frequencies can be confused for selection. They sort of it looks like there's selection when it's really just these heavy tails. And it has to do with the more dominant strain sort of more deeply sampling that tail. There's close connections to stat mech here too. So, you might think of the sum as kind of like the partition function. These ratios are kind of like the Gibbs measure. So in terms of the relationship between those two concepts mathematically, and I think in terms of the recent efforts and sort of the ongoing work is a lot of interest in understanding selection in the context of these heavy-tailed offspring models. Okay, I'll skip that. There's kind of more examples. So that was about growth and how sort of we encounter heavy tails in the context of growing populations, again, because we're amplifying these rare events. There's another class of problems I'll talk about, which is really more about timing and search. And the first two talks were a little bit more of this flavor, although actually they don't fit squarely into my examples here, which is good. That's kind of, I like how it worked out that way. Yeah. 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 Yeah, so I mean, I'm talking about the genealogy. So like, I guess the best way to explain is if I go back to this picture, I run this population dynamics. Now let's say I sample some number of individuals from the end, and I'm tracing back their genealogy. I'm just following the branches back, and that eventually, if I follow it far enough, they're all going to coalesce, and that I can then redraw that tree more nicely. And I would get something like this in a large population over a long time. So that's like how it would be constructed from those. That's literally how I made these figures. Is I just took those simulations. Yeah. Yeah. When I talk about the coalescent, I'm talking about this tree. Technically speaking, it's a continuous time Markov chain on the space of partitions of the natural numbers. So you start with all singletons, and then you have the transition probabilities merge the groupings of those. So a lot of problems in biology involve like something having to find something, like a molecule having to find something. So one example is a transcription factor. These are things that bind and unbind to the DNA and control whether it's transcribed or not. And they have to find the DNA to do that. And there's also examples in neuroscience. This is a mRNA localizing in dendrites. So it's super important that this mRNA gets localized into certain regions for the function of these neurons for reasons I won't get into. But ultimately, these are problems that involve particles need to be in a certain region, or they need to bind to a certain thing, and it needs to happen on certain timescales for cells to function. Again, we saw some of this in the first two talks. And we like to model things by diffusion because diffusion is super nice. But as I learned, as they talk about in this review, and as talked about in a lot of other books and reviews, is diffusion is too slow. So to give an example again that comes from this review paper, I'm sure it's given elsewhere, is if you consider a particle diffusing on 0L, interval 0L, and you define the first passage time to the end of that interval, and you ask, okay, what's the average time it's going to take to get there? That's going to be L squared over D. You can get that from dimensional analysis. That's going to give you 10 days to diffuse a length of an axon. So that's far too slow for a lot of the things we need to do in cells. And the way cells get around this is they actively transport things. So they have various ways of taking a particle and sort of shuttling it between two regions. And so an example is these transcription factors can attach the DNA, slide along the DNA, and that makes them essentially move much faster than they would if they were just diffusing. And then there's these, we heard about these like microtubules a little earlier. So there's these filaments in cells, and they're these like little machines. They're kind of like the highways in the trucks of cellular physiology that bind to the microtubules. They're proteins that bind to the microtubules and sort of shuttle things along them. And again, they're not diffusing, there's active transport in these processes. And this motivates the study of these first passage times for kind of different types of processes that are not diffusion, and it leads to all sorts of interesting mathematical problems because they're not parabolic PDEs, they're hyperbolic PDEs, or they're some kind of hybrid PDE. I won't get into this too much, just in the interest of time, but so there's been motivated by these questions, there's kind of a massive amount of effort to understand first passage times for different types of stochastic processes, and that is some form of an extremal process because you're looking at what's the first time. But you can get even more extreme, and David Holkman and co-authors have argued that for a lot of these problems, you're dealing with many, many particles. You're trying, another mechanism cells have to speed up these times is to pour many, many particles into a system where you need a particle to, say, bind to a receptor on a certain time scale. And this leads to even more extreme problem where you're interested in sort of the extremal first passage times of many, many particles. And here's just showing another example where you have neurotransmitters. Many, many of them are released, but it's the first one that binds to this NMDA receptor that gets amplified, right? So if you want to understand this process quantitatively, you want to look at the extremal statistics there. And there's been a lot of math, and I won't talk about it, but there's been a lot of attention to these problems and the extremal statistics in different limits. Often you're dealing with sort of double limits where you're looking at many particles, a small target, and the asymptotics there, involves a lot of PDE asymptotics that in the mathematics literature has kind of taken on a life of its own. So this is another area where we're seeing kind of really fundamental biological problems sort of leading to a new sort of mathematical subfield and then the feeding back into each other. And this I won't talk about. And then the last class of problems I want to discuss are those which involve, I call this disordered systems, but basically problems that involve systems that have many, many fixed points or many, many favorable configurations. So example would be an ecosystem where you have many different species. I mean, there's 6, but this is a cartoon. We have many, many species and many, many different, in this case, there are these different metabolic byproducts that they produce, and they're sort of feeding on them and producing them. And you can imagine that this system, there's maybe many, many ways I can sort of make this system in a stable fixed point. And then you have also in evolution situations where you have sort of a fitness landscape. So fitness is a function of the genotype, which has many, many, many peaks. It was very rugged, okay? And these ultimately are problems, I mean, you can kind of see why they're problems about extremes, because they're problems about trying to find, say, in the fitness case, the maximum of this landscape. But so why are they problems of extremal statistics? And that has to do with the approach that we take in physics and math to understand these sort of very complicated systems with many, many parameters, because you're going to have parameters for each of these interactions. And what is done is we tend to think about what's the typical system look like? What would be a typical system instead of trying to find those parameters exactly because we can't know them. We think about what's the typical system look like. And so, for example, in the ecosystem model, I'm just writing down one example of equations that you can use to describe a ecological system where you have the number of cells, Ni is like the number of species I, and you have, the important thing here is that you have these terms alpha which describe the interactions basically between the species. This is an example of a consumer resource model, type model. Actually, this is actually a reduction of a consumer resource model to more of a Lotka-Volterra model, but that's a bit beside the point. But So what we would do is, instead of trying to find some values of these alphas, because we know they're kind of all over the place in real life, we don't know what they are, we just take them to be normally distributed. This is kind of an idea inherited from the physical disordered systems. And the same idea, if we have a fitness landscape, you can imagine that fitness is a function of the different genotypes. This could be like 0 or 1, depending on whether there's a mutation on the ith site of the genome. And the fitness is going to depend maybe on, I've just written one example of a landscape you could write down. It's going to depend on the products of the genome. So how the genomes interact, how changing a mutation on site I is going to affect the fitness is going to depend on what the other sites is on the other sites through these interaction coefficients, and we take those to be random. And this is actually, in physics, they call this the Sherrington-Kirkpatrick model. But there's sort of different questions that would be asked about it in the context of biology. So, see how I'm doing for time. So, basically, in physics, the tools that are used to study these sorts of systems, these complex systems with many, many interactions, come from disordered systems and physical disordered systems. And there's this idea of replica symmetry breaking, which is ultimately an extremal phenomenon. And so here I'm showing, this is like a little bit of a detour into physics, but here I'm showing the magnetic susceptibility of a copper alloy. So it has, it's copper and it has some magnesium in it, which at low temperatures is kind of randomly distributed throughout the alloy. And it turns out what people realize is that there's two different magnetic susceptibilities you can look at depending on whether you're looking at the magnetic susceptibility in a given configuration. So like you're freezing the system in a disordered configuration, you're looking at the magnetic susceptibility, that's the lower one, or whether you're doing this over an average over different realizations of this freezing process. So they call it the FC and ZFC magnetization. And the kind of study of these phenomenon has led to a lot of sort of a toolbox for dealing with these kind of systems where you have a complex system with many, many favorable configurations. And these mathematically, the distinction between these two magnetizations, if we're thinking about the free energy, so free energy is about essentially the growth rate of the partition function, but in a disordered system like these, we need to take an average. And this distinction basically comes from where do you put the average before or after taking the log when you're calculating the growth rate. And this idea also appears, just as a side note, it appears in biology in sort of this idea of arithmetic versus geometric fitness. And essentially the distinction between these two things, this is kind of to circle back to the beginning and what I was talking about, kind of the central limit theorem and whether we have a law of large numbers in these transitions in power law sums, the distinction between these two free energies, the quenched and annealed, depending on whether you put the expectation outside or inside the log, is basically related to whether you have a law of large numbers for this sum, whether it converges in a certain sense to its average or not in the large system size limit. So this actually, the techniques, this is just one example where this is a guy, Bunnen's work is constructing a phase diagram for one of these ecological models. And basically he uses this cavity method, which comes from this disordered system. So these have found many applications in biology. And again, it's ultimately it's about extremal statistics and even sort of related to these simpler things I talked about at the beginning of the talk. And the last thing I want to say just about this part of the talk, or just about these sort of disordered systems, is that you can really tie it all the way back to the beginning of the talk. If you think about, I'll skip some of the sort of technical details here, but if you think about the kind of the most disordered system you can think about, the highest, the most rugged landscape, well, that would be just taking all the energy levels, all the different energy levels in my system, or if we're thinking about evolution, all of the different fitnesses to be just independent, identically distributed random variables. Forget about the alphas. right? Those alphas, those interactions, they're just random. So instead of thinking about the randomness in the alphas, we can just think about the randomness in the fitnesses, and then we just get some random landscape. And in the limit, where that's super, super random, they're all uncorrelated, the partition function is just an IID sum, and we're all the way back to the beginning of the talk, where I'm just trying to figure out what happens in the limit to some IID sum in the same kind of techniques that Janjenko and Kolmogorov used to figure out those limit laws for IID sums. You can apply to these sums, and that was work by Benarus and co-authors, and Bouvier and Rel, so people who have all kind of connected these dots in the literature. And in evolution, because we're talking about biology, in evolution they call this the house of cards model. The idea is that it's such a rugged landscape. You make one mutation, boom, the whole house of cards comes crumbling down. You have a completely new fitness. So we've come full circle. There's a lot of things I didn't mention in this talk that do fall under this umbrella of extremal statistics. So I didn't really talk about machine learning, which is widespread in biology. And this is also closely related, like questions about machine learning algorithms and when they work is closely related to extremal statistics. This is going back to Hopfield. And there's also sort of more questions about like classical statistic techniques like Markov chain Monte Carlo, which are used in biological applications. It's not really extremal statistics in biology, more in data analysis of biology, I guess you could say. And I also didn't say anything about random matrix theory, but that's also something that sort of, there's a lot of the questions I talk about mathematically have sort of analogs in random matrix theory, things about like ordering of eigenvalues and extreme eigenvalues. So hopefully that just gives you a sense of the scope of this field and how the mathematics and the biology have kind of interacted with each other. Thank you everyone for being here. Obviously thank Ariel and Christoph for organizing this with me. It's been a lot of fun, and it's going to be a great week. We already got to a good start. We saw some Gumbel distributions, so it's a good way to start the day. And I also thank the Clore Center for Biological Physics at the Weizmann Institute for, they provided very generously the support for this. So thank you.